Good afternoon everyone. Myself Bhavana, DL in Microbiology from TTWRDC DEVARAKONDA. Today we are going to discuss about a topic called Hybridoma technology. Before going into the topic, let us know what are monoclonal antibodies and polyclonal antibodies. So we all know that whenever our body encounters with an antigen, our immune system stimulates and produces antibodies to fight against those antigens. So antibodies produced from a single clone is called as a monoclonal antibody, and antibodies produced from multiple clones are called as a polyclonal antibodies, and the process of production of these monoclonal antibodies is called as a hybridoma technology. This hybridoma technology was first discovered by Kobler and Milstein in the year 1975, and for which they got Nobel Prize in the year 1984. So actually, what is hybridoma technology? Means in this technology we are fusing B cell, that is plasma cell originated B cell, with myeloma cell. Myeloma cell is also called as a cancer cell. So what is the principle behind this hybridoma technology? That means B cells are short-lived, but they continuously produce antibodies. Myeloma cells or cancer cells, it has a capacity to grow continuously. So if we mix these two cells, we will get a hybrid cell. You are calling it as a hybridoma cell. In this cell, we have the property of both B cells and myeloma cells, that is antibody production capacity from the B cell and immortality, long-livedness from the myeloma cell or the cancer cell. So the process of production of these hybridoma cells or hybrid cells is called as a hybridoma technology. So what is the process behind this hybridoma technology? Means first you have to immunize the experimental animal with antigen. So for which kind of antibodies need to be produced, such antigen is to be taken and it is given intravenously to that experimental animal. You are immunizing the animal so that its spleen gets activated and the B cells, they get activated and they produce plasma cells. These plasma cells in turn, they will produce monoclonal antibodies. Next, you have to separate these monoclonal antibodies. In the next step, you have to separate myeloma cells from the bone marrow. Myeloma cells are nothing but the cancer cells which has a capacity for the regrowth, for the long-livedness. So these myeloma cells are separated from a bone marrow. In the next step, you need to fuse both B cells, that is plasma cells, and the myeloma cells in the presence of PEG, polyethylene glycol. It favors the fusion of both the cells. In the next step, we will get 5 kinds of cells like fused B cells, unfused B cells, fused myeloma cells, unfused myeloma cells, and hybridoma cells. In these 5 kinds of cells, you need to separate only hybridoma cells because we are interested only in these hybridoma cells. Remaining cells should be isolated. So you have to isolate hybridoma cells from all these cells. So these hybridoma cells can be cultured in a medium called hat medium. So before going into this topic, let us know a small point. For the nucleotide synthesis, any cell, it follows 2 pathways like de novo pathway and salvage pathway. What happens if this hybridoma cell isolation? So you are culturing the hybridoma cells in the hat medium. Hat medium is nothing but hypoxanthine, aminopterin and thymidine. This aminopterin, which is present in the hat medium, it is blocking the dihydrofolate enzyme, which is necessary for the de novo pathway. So de novo pathway cannot occur in this hat medium. Next, salvage pathway. For the salvage pathway, you require an enzyme called HGPRT. That means hypoxanthine guanine phosphoribosyl transferase. This enzyme is required for the synthesis of nucleotides. But in these cells, B cells is having this enzyme. So B cells can be divided, can be multiplied. In myeloma cells, this enzyme is absent. Myeloma cells is negative of this enzyme. So myeloma cells cannot grow in this hat medium. B cells can be grown in this hard medium, but B cells are short-lived, so those cells get died. As myeloma cells don't have this enzyme, they cannot synthesize the nucleotides by this salvage pathway, so myeloma cells will not grow further. Coming to the hybridoma cell or hybrid cell, in this cell you will be having the capacity of B cells. That means this HDPRT enzyme is present. So this cell, this hybridoma cell, it grows in the hard medium, and finally we will get hybridoma cells only, remaining all the unfused B cells, unfused myeloma cells, fused B cells and fused myeloma cells all get died in this hard medium. Only this hard medium is selective for the culturing of this hybridoma cells. So after the culturing of this hybridoma cells, they are screened by using different immunological techniques like ELISA. After isolation, it can be commercially sent into the markets. So what are the applications of these monoclonal antibodies? These monoclonal antibodies, they are responsible of a serological test or diagnosis process or preventing diseases also. So nowadays we are performing blood tests. Those are nothing but monoclonal antibodies. So for the identification of the blood groups or for the pregnancy diagnosis, we are using this monoclonal antibodies. And also monoclonal antibodies can be useful for the interferon purification. And finally, these monoclonal antibodies, they can be used for the cancer treatment also. This is all regarding monoclonal antibodies and hybridoma technology. Thank you.